Good morning, students. Here we come with 8th chapter of class 10. That is cell biology and biotechnology. So the 10th chapter, 8th chapter, that is cell biology and biotechnology. What is cell? Cell is what? What is cell? Cell is the structural functional unit of living organism. So you might have studied in the previous standard what are cells. So cells are what? Cell is the structure and functional unit of living organism. Every living organisms are made up of cell, like we human beings. We are also made up of cell, different minute cells. Like many cells, when come together, they form the tissue. When many tissues they come together, they form the organ. When many organs come together, they form the organ system. Our body, which is made up of different organs like liver, kidney, heart, brain. So what is that? That is the organ system. So similarly, the cell is what structural and functional unit of living organism. Every organisms are made up of cell. So there are many different types of cells. Like stem cells are there. Again, in your blood, there are two types of cells. RBCs, that is red blood cells. We also call as red blood corpuscles. And WBCs. WBCs is what? White blood cells. Or we can see as white blood corpuscles. Okay. So cells, who has invented cell? The Robert Hooke. He was the first to do the invention of cell. He had in a microscope he had found just a small like structure. So he was noticed that. So he found a honeycomb like structure, and then he named the as a cellula. Cellula means a honeycomb like structure. That is the cell. Now, there are different cells like RBCs, WBCs, stem cells. So, stem cells are what? These stem cells are these are special types. These stem cells are special types of cells. These are present in our body, but these are not unicellular. These are multicellular organisms. So they the main importance like they play important role in wound healing. Whenever you get injured, so any maybe accident or any case, whenever you fall down, so you get injury. So sometimes in that case, you will heal that part very fast. Why? Because that or the same cells are well developed. If you see in the diabetic patients, so the injury, whenever they got the injury, so they don't get the injury fast. Okay, so they require time as compared to the normal person. So stem cell play important role in the wound healing. So now we will move to the next part. That is what the tissue culture. How these cells are cultivated artificially. So here that is ex vivo. Ex vivo means what? In artificial way. Whereas in vivo means what? In our condition, means in normal condition, like we are growing the different plants on in agriculture. So here we will see tissue culture. How the tissue is grown in ex vivo. Ex vivo. Ex vivo means in artificial way. So here I have took the source plant. It may be any source plant like carrots, you can say carrot. So I have taken the coconut plant and I have taken a source of a plant that, and then I have cut the bulb corn of a source. Means I have cut that and I have done the primary treatment here. First is what the primary treatment. In this primary treatment is done in lab condition, we can say. There in microbiology labs, there are the different waters we require for growing in artificial way. So these are the waters that is grown in artificial way. The plants are grown in artificial way. So that is we call as here. There we have put the callus. This source plant we have cultivated here in the water. So this is called the primary treatment. Like we requires in the normal plant, we requires the different nutrients, macronutrients. So in the artificial way also, we have on the medium, we have to put the different nutrients, macronutrients to grow. So first that is the reproduction and multiplication. Whenever we will put this plant in the water, we will found what that is rooting and shooting. Means rooting means what the roots will develop here, and shooting means what? Shoots means the stem. Stem means the leaf part. Roots will develop here in artificial way, in the medium. So that process is known as rooting and shooting in artificial way. We are not cultivating in normal area, in open area, but we are cultivating in a lab condition. So the next that is primary hardening. So primary hardening means what? After the cultivation, these plants are remove out safely, and then it is cultivated, then it is grown in a some small pots. So that is called for a primary hardening. That is, you might have seen in the nurseries or any other place that we got such plants, small plants that are called the which we are cultivated on in the open space, or we use for planting in our pots. So many of the agriculture, they depend on this process for reproduction and the multiplication. So in this varieties, if you see in the Jalgaon, the tissue culture lab is there. So at that in this tissue culture, so mainly the various plants like banana plant, coconuts. So these are grown in the artificial way, and so as this culture in the artificial way, so here the reproduction and the multiplication will grow in the past. Within a small period, we will get number of plants growth, and we will get number of varieties, and not the varieties, but we will get the increase, and again the new varieties of the plants we can cultivate it by the ex vivo plants, by ex vivo method. That is the tissue culture method. So just we have to put the small part. It can be a call as a bulk call or a source of a plant. Then we have to do the primary treatment in the lab condition in a bottle, in artificial bottle. We can also use the petri plate. Petri plate, that is the glass plates, and these are the culture bottles, we can say, and we have to add the medium there, and on that medium we can grow artificially the plants. After growing such artificial plants, then they show the rooting and shooting process. Then after that there is the primary hardening. After that these plants are removed safely, and then it is sown in the pots in the soil. So after that soil, it is now we can use these plants for our agriculture. So in next part we will see what are its application. If you like my video, please like and subscribe the channel, and don't forget to share with your friends. Thank you for listening. Have a nice day. Carry on. बंद नहीं कर देगा ये टॉपिक बुकले तो क्या मुझे वीडियो कर करके बोल रहे हो बुकले पढ़ा चैनल के आप लोग के ऊपर नहीं देखना है और क्लिक नहीं होता अच्छा तो क्लियर हो रहा है ना आधा घंटा दे सकते हैं ना नहीं हो गया तो बंद क्यों Okay. So the next we will come with the what is biotechnology. So here the vast branch that is the biotechnology. So here we can say that the stem cells these are artificially grown with the help of biotechnology. So nowadays in such a pandemic situation you are facing, so you see the different biotechnology lab, virology labs. So here they have made the different efforts they are cultivating for making the vaccination, you can say. So as the cell have the cell division. So means cell division means what? One cell can is converted into two cells. Then after two cells that is again divided into four cells. That four cell again divided into eight cells. Again eight cell that is divided into the 16 cell. This 16 cell then again divided into 32 cells. Then 64. The number of multiplication, the number of division is more and more. So here the same case that is applied in viruses also. So in this COVID-19 we are facing, so the viruses that grow in a minute part, we can say within a second that grow into 32 or 64. So because of the cell division that took place in cell also and virus also. So for this, the biotechnology play important role. So biotechnology means what? Bio means biological and technology means the technical part. So here the technical part like computers are used here. So okay. So we can say that in biotechnology, the same step research is revolutionary after the cloning. So this step cloning has a potential and bringing the fundamental changes in the medicinal science. So just now I explained about the medicinal science that is about the viruses. Many of these companies like biotechnology, nanotechnology, they are inventing new medicines, they are inventing new viruses. So that is depending on the two types of cell. First, that is embryonic cell. Second, that is adult cell state. So we will see what are the embryonic cells, stem cell. So embryonic stem cell means what? Division of the zygote and that starts nearby and they convert into the embryo. Cells of embryo undergo the repeated mitotic division. So in previous standard you have might learn about mitotic meiosis. So the zygote, the first development is the zygote, and that zygote is converted into the embryo, and the cells of embryo undergo repeated division. Just like I said about the how the cell is divided into 8, then 8 it is divided into 16. 16 is again divided into 30. 32 is divided into 64, and number of the cells we can count. So this is process applicable for both embryonic cell also. So the cell differentiation start from the 14 and the cells of the different organ like oocyte. Oocyte means or you can say bone cell that these are present in bone marrow. So hepatocytes, hepa means what liver that is related to the liver. These cells are found in liver. And neurons. Neurons means you have might studied about the neurons, nephrons. So this neuron that is related to the brain. Nervous system. Neurons are found due to the differentiation. So the embryonic state before the differentiation are called embryonic stem cell. So there are different types of cells. In 222 different types of cells of human body are formed from single type of cell embryonic state. So the stem cells are the primary and undifferentiated with the self-multiplying and ability to have a parent cell and different human cell. So these cells require 14 days. The duration time of this growing such human cell that is the same cell, it requires 14 days. The first duration that is 5 to 7 days. During 5 to 7 days, the culture can be certain biochemical stimulus in laboratories. Just like we have seen in the in vivo, so these cells are also cultivated in the laboratories by artificial way. Whereas, as for the stimulus, they are transformed themselves into desired types of cell thereby tissue, and finally into the organs. So, without the second, that is adult stem cell. Adult. So, from the name you can say that adult means what? The stem cell can be obtained from the body of the adult person. It may be your human beings, you can say. These are the three main sources of the stem cells of the body of the adult person. The stem cell can be formed from the red bone marrow. So red bone marrow that is present in our disease. Again, adipose connective tissue. So in the different previous standard, you have studied about the tissue, the types of tissue, connective tissue, epithelial tissue. So this that is that is adipose connective tissue. So connective tissue means that the body parts are connected. Just we have the legs, we have hands, the connective tissues. These are present. So stem cells are formed obtained from the cord of blood and emanate after the blood. So in the next part, we will see the uses of the stem cell. So thank you for listening me. Have a good day. If you like, and you also subscribe to your friends. Subscribe our channel and share with your friends. Have a good day.